I would now like to introduce our first speaker. As I said, Ann Poduri is joining us here from Harvard Boston Children's. That's a lofty handle, right? Harvard is a university and it's a brand. But Ann is not like that. Ann is a scientist who's in it for the love of learning and the love of science. And it's very important and symbolic that Ann is here, not only because she's come from far away to join with all of us, but because from speaking to Ann, it's Ann's premise, it's Ann's belief that we have to deal with all genetic epilepsies. None are more equal than others. They are all equally important. And as we come on in. Anybody at any time today? Remember, family, come on in. As we deal with all of our disorders, we help everyone in this room. Sometimes it's going to seem like somebody might be ahead. Gavin's right, it's not a competition. It is not a competition. It is working with our family, right? And there's not a race except to get all of our kids better, because we all know what it's like, and we all know how hard it was to get here today. So welcome, we're so happy you're here. So may I allow Ann Poduri to start off, and you'll get questions after. Ann, please, warm welcome.
Thank you so much, Sarah.
Is that all right if I sit here for a minute? Is that all right?
Okay. So I'm really honored to be here, and I thank Sarah and the entire GETA group, and also Professor Ingrid Scheffer, who's really inspired me in many of my endeavors, and it's really sort of was my connection to Sarah, so that's really been a wonderful connection, and I hope to keep cultivating it. So I call this Precision Medicine for Genetic Epilepsy, Small Steps Toward a Big Vision, and I do think it is a big vision, and it's a tall task. And a lot of us are told early in our careers that we're sort of asking for too much and being too aspirational. But the truth is that if we don't start taking small steps to get there, we'll never get there. And if we start taking small steps together, we actually can be on our way. So I'll try not to qualify too much. I don't want to seem pessimistic, but I also want us to be realistic, as Sarah was, that we're on our way, and it's going to be a long journey. And the way to journey is to do that together. I just want to cover some basic definitions. Most of you know what epilepsy means and all of those things, but I just want to do a little bit of epidemiology and definitions and talk very briefly about our history in medicine and science with epilepsy genetics. And then I'll talk a bit about traditional paradigms of how we practice epilepsy and epilepsy genetics versus translational paradigms of trying to integrate our precision medicine aspirations into our everyday practice. And then I'll talk for a little while about the model systems for genetic epilepsy. How do we want to model this disorder and set of disorders? And then at the end about some treatments and how we're trying to push the envelope beyond where we are today. So just some basic definitions. What is a seizure? A seizure can be thought of as an abnormally synchronized paroxysmal electrical event in the brain. So what does that mean? Suddenly things are going off that shouldn't be, and they're all organized. And it typically, but doesn't always, result in an outward clinical manifestation. So if my right motor cortex is having an excess of synchronized firing, my left body might have rhythmic jerking, and so on and so forth. So where the seizure happens in the brain dictates what it looks like on the outside. And as many of you probably know, you can also have electrical abnormalities going on in your EEGs and going on in your brain that are not necessarily showing with anything outward. And it turns out that about 1 in 10 people, all comers, will have a seizure. So seizures themselves are not that rare. And one of the themes I'll try to pick up on is that while we are dealing with individually rare entities, we're dealing with things that actually affect many, many, many people. And so that's important, I think, for you all to know, because everything that comes out of the research on your children's condition will actually have impact, I really believe, on a wide group of individuals. So what is epilepsy? Epilepsy is more than one unprovoked seizure, or the little asterisk is the tendency to have more than one unprovoked seizure. And as if that weren't enough, we are often dealing with epilepsy syndromes where we can have not only epilepsy, but specific ages of onset, types of seizures, developmental disabilities, and sometimes specific EEG findings. Alternately, we might think of an epilepsy syndrome as a constellation of conditions related to epilepsy, sometimes gene-defined, including developmental delay, intellectual disability, autism, hypotonia, movement disorders, and so on. And I'll highlight a couple of features here by underlining them, just to say that we are focusing a lot on the epilepsy, but we have to remember that there's a whole constellation of conditions, and sometimes these other conditions on a day-to-day basis are actually more impacting the quality of life of the children and families. And then particularly here, because it is GETA, we're talking specifically about gene-defined epilepsies. So our traditional framework, and this is sort of a stylized cartoon that several of us put together when we were grouping ourselves as an EPI-PM community, epilepsy precision medicine community of physicians and scientists. And what this part depicts is that we have to do something in our clinic. So on Tuesday afternoon when I'm in clinic, I might make a diagnosis of a developmental epileptic encephalopathy, for example, do a genetic assessment if I can. But then what do I do in terms of treatment of the epilepsy? I use the same drugs that have been used for decades and maybe some of the newer drugs, but I'm really taking an empiric approach to symptom management. I'm not really taking into account that genetic diagnosis most of the time. And often when we're seeing a patient, we're still doing the genetic evaluation. We don't have the answer yet, and so we have to pick something that kind of makes sense based on the type of seizures and based on the age and side effect profile, but it's not particularly scientific in its approach. So the problem really is, even though we've had this great explosion of genetic knowledge, is that we have a growing list of drugs, more and more drugs, and yet we still have a third of our patients overall refractory to medications. And then when you were talking about the rare early onset genetic epilepsies, the vast majority have refractory epilepsy. And so despite a lot of progress in terms of the numbers of drugs available, we still really don't have specific drugs for most of our genetic conditions, and we still really haven't made a dent in how many of our patients respond to the group of drugs. So the promise of a solution to that problem comes through what we have now heard as a buzzword of precision medicine. And this is really encapsulated nicely as a new approach where research technology and policies can empower patients, researchers, and providers to work together toward the development of individualized care. When I talk to some of my family members who are not doctors and not researchers and tell them what I'm doing, they say, well, isn't that what you'd want for all of healthcare, for any condition? And of course it is. And it seems like in 2018 we shouldn't have to say that. But in fact, most of what we do in medicine is a practice, and it's a practice based on years and decades of observation and some clinical trials and some clinical experience. But it is still a practice that we need to drive with more science. So the science here comes really first from epilepsy genetics. And so just by way of introduction, this is just a description of how we used to think about things. This is actually from here, from Sam Berkovic and colleagues, and a depiction from the 70s of how we really thought of the causes of epilepsy. That you could have infectious causes, you could have a tumor, stroke, et cetera, but the vast majority were called idiopathic. And that's how I learned about them. And then over time, they were termed presumed genetic. And this is because of some family studies, which is a small number of cases, but many of them from Australia and described really first here. But many, many different groups of patients who were grouped together with the same types of epilepsy syndromes. And this really led to the belief that there must be common causes among these syndromes. So if we then fast forward to the modern paradigm, it turns out that much of this has been filled in now. It's not all sort of a straightforward single gene epilepsy, but really what we're here to talk about mostly is single gene epilepsies. They can be familial, they're often de novo, so only the child affected has the actual change detectable, the parents don't. And then there's a whole host of things that we still haven't really cracked. So what do we do about modifiers and susceptibility alleles? Why can the same mutation in two different children have two very different effects? And maybe it's because of other genetic factors or other susceptibility factors. So there's a lot we know, and there's a lot we're still trying to understand. So how can we then go from that traditional framework of here's a clinical diagnosis, I'm going to try to make a genetic change knowable and diagnosable, but then still use the same treatment? How do we take that beyond just that inner circle? And the way that I think of this is sort of expanding my Tuesday clinic experience to the rest of my life experience, which is on the research side of things. And there we have not just clinical diagnoses one patient at a time, but clinical registries, which many of you are participating in and actually helping us to develop. Expert review of variants. So when we see a change, is it really an important change? Is it changing the protein or is it not? And several of us have grouped together through an NIH and CURE collaborative called the Epilepsy Genetics Initiative to do that together. And then finally, how do we take all of these observations, which are at the core clinical observations that we might make, one doctor, one family in a room. We're trying to make them not just single observations, but collective observations, and at some point really bring a level of clinical rigor and clinical research rigor to these observations, and to take what we know from the clinic over to the bench research side. And then this is really where you'll hear more from some of the talks later on, where really, really deep functional data has to be pursued. And so there are people who've been spending their careers on the very, very channels and the very, very proteins in the synapse that are applicable to your children. And that's miraculous, because we now have this wealth of clinical knowledge and clinical science that we can partner with that to really make some understanding of what's important to study, what's most important to prioritize, what aspects should we prioritize, and how can we then take all of that to drive clinical trials, which we'll need for real precision treatment. And we've made great headway here and a lot of headway here. There's a fabulous science going on, which you'll hear about, and really takes a whole enterprise unto itself. Really, this aspect of the translation is where we need to really push the hardest, I think, in the next decade, to bring these discoveries from the bench back to the patients who have motivated the science. So with that in mind, what are we seeking in epilepsy models? And I'll bring this back when we talk about what we're talking about with what do we want to treat. Again, this comes from the importance of phenotyping, of doing rigorous, detailed phenotyping and putting observations together in a very rigorous scientific way. Because when we think about what we're trying to study, that's also what we want to treat. And the better we understand that and the more we can get at its core features, the better we can do that systematically. So are we talking about acquired epilepsy versus genetic epilepsy? Many of the animal models of epilepsy until recently were acquired epilepsy models where we're studying induced seizures, and my lab does some of that as well. There's a role for that. But really, when we're talking about genetic epilepsies, and we have genetic tools among us now in our labs, why not study those specific genes in our models? Likewise, provoked seizures versus spontaneous seizures. Children with epilepsy have spontaneous seizures, or the tendency to recurrent spontaneous seizures. Many of our animal models are actually looking at provoked seizures, provoking with chemoconvulsants, things that make the animals seize. And that can be a valuable research tool, but really that's not as applicable as the spontaneously seizing model. So that's sort of what we're after if we can. What about the other features, such as the intellectual issues, the mood, and the behavioral issues? Those are all things to think about, but depending on how complex our model, we may or may not be able to get there. But I think we can at least hone in on some basic goals for how we want to study genetic epilepsies. So we want a model that has sufficient homology to humans. So what do I mean by that? The DNA of that model system should be close to ours, if it's not identical to ours, to be able to translate back and forth between systems. The real features for me that are important is there a relevant phenotype possible? Meaning, what is it that we can actually model? So to review from earlier, the core epilepsy features are the seizures and the electrical changes that we might see on an electroencephalogram in a human. Can we model that in animals or in cell culture systems, even with seizure-like phenomena or electrophysiological abnormalities? And that's an important question. Certainly, we want genetic tractability. What does that mean? Can we actually introduce changes that really model the genes we want to look for? And we certainly can. There have been a variety of techniques over the last decades, and really the one that's been in use a lot in the last few years is the CRISPR-Cas9 system. I won't go into the details, but really this is a nice system that lets us introduce specific changes or to be able to remove the function of specific genes in a very focused way. And so that's been quite a great tool of this decade. And then finally, if we're talking about translational and drug screening studies, which you'll hear more about from Steve, I'm sure we want to talk about bioavailability and throughput. So if I'm going to study an animal model, it would be nice if I could actually give it a drug that it can absorb and can get into its brain if we want to study epilepsy. And we'd like to be able to do this fast enough that it doesn't take us 10 years to get through every model. So this is a very, very biased comparison of models now, which is really sort of a justification for why my lab is using the zebrafish system. There may be some dissent in the room about whether that's the best model system, and that's okay. There are lots of different model systems, and you'll hear about a lot of them. We happen to be working on the zebrafish system, so I just wanted to provide a little bit of context for that, and you'll hear about other ones later on. So what I've mapped out here is the homology in terms of DNA. You can see there's very different numbers of neurons that we all have, brain weights based on our size and proportion of brain to body size, and then genetic homology. So if we start with ourselves at the top, we're 100% homologous with each other, sort of. We won't get into the details. In fact, we're not. But if we look at other species, a mouse, for example, that's very commonly studied and in many ways considered kind of the gold standard in terms of what do we need to go from an animal model to translating. We've got about 75 to 80% homology. So the zebrafish is actually not that far behind. It's at 70% homology. Some genes are very much similar. And what I mean by homology is you line up the DNA sequence of us and of a certain gene, and you line up the DNA sequence of the zebrafish. In some cases, they're very similar, 80 or 90% similar. In some cases, they're not, 40% similar. So we're sort of inclined to study the ones in this system that are more similar to us. Depending on how close to human you are, you can sort of rank them this way genetically, and certainly the cost and the time, and invariably, eventually, the ethical issues come into play as we get into primates, for example. The ethical issues become a big issue. And so really there's a variety of factors that come into play in terms of choosing your model. There's a lot of what we call throughput advantages to the zebrafish, and I won't get into all of the details, but because we've got sufficient homology to go forward, the other thing that's quite handy is that it's actually quite easy and relatively inexpensive compared to rodents to breed them. And they grow very quickly. So from fertilization to 5 days, you actually can have an organism that can display seizure-like behavior, which I'll show you. And you also then, within a few weeks, can look at a whole range of behaviors that I won't go into, but behavioral types of phenotypes. So we actually think this is a nice model with reasonable throughput. The other thing is if you have a clutch of eggs, unlike just a handful of animals, you actually can have 100 or even 200 larvae at a time. And so that gives you the ability to have a lot of numbers and to be able to reproduce experiments, which we find very important in terms of our validity and reproducibility. But do larval zebrafish really have seizures? I keep saying we need to model things that are important, and so I would argue that they do, and really nice work from Scott Baraban at UCSF has given some really elegant demonstration of this. So I'm just going to show you what the fish look like. This is, imagine a couple millimeter fish. In addition, this is what it would normally look like. They take these little strokes. They have this sort of beat and glide swim mechanism, so they'll do a beat of their fins, and then they'll have a little gliding mechanism. And if you give them a proconvulsant, and this is a little bit traumatic actually, my kids don't like to see this, but they actually do have this very, very aggressive, forced, tonic activity where they're swimming around, and then at the end they kind of lay on their sides. And so we can look at that one fish at a time in a little dish. We can actually do motion tracking with some very, very simple technology that's just countertop. It's about this big. Again, one well at a time. So there you can see the distance that's been traveled there by this fish much more than the other one. But one fish at a time isn't so efficient, and we're getting 100 or so at a time. So we can put them into these 96 well plates. And I like to walk by and say, I think, look, let's see, I think maybe B2 looks like it's swimming pretty fast. And that's not any more efficient than looking at them at one at a time, but we can also heat map these and generate a readout of the distance traveled, the velocity that they're traveling, the maximum velocity that they reach. And it turns out that there are some very reproducible core features. And so when we've done this and we've tried to get the computer to read this, and I'm not a big believer in big machine learning and so on, but the young people in the lab are. They were able to do this to such a degree that we actually had really quite not only reproducible results when we ran the algorithm multiple times on multiple plates of fish, but we had 4 blinded individuals rating are the core features met just from a phenotypic perspective, and the agreement was actually quite spectacular. So we'll at some point report this when we have other findings to report, but it really, when we look at one well at a time, we can split out the seizing fish from the non-seizing fish very nicely. So we can actually phenotype in about 10 minutes 96 fish at a time, which is great. So in kids, we evaluate kids with video EEG sometimes, and then we evaluate them with EEG and video, and you send us videos and all those things. So we can't do video EEG quite yet in fish. We just don't have the way to put the two together. We can, and we've tried to do various forms of EEG in these little larval fish. This is looking at a microelectrode array that are often used in cell cultures that people use for their cell work. And this is something we just put on the head of the zebrafish, and we were able to get reasonably reproducible readouts, but at extremely low throughput. Scott Baraban at UCSF has been using a single electrode method, and we've been actually trying to look at that as well, and to use that as a basically validation that when we say a fish is having a seizure, that it actually has some abnormal spikes and maybe even some electrographic seizures. So that's work in progress that we're working on. In terms of the genetic tractability, this is where it's actually quite easy. And any of you, I would say, in a couple of weeks could learn how to do this. The eggs are produced, and you can inject with a little injector these constructs that will change the DNA of a specific target, and will target the gene that we want to target. And what they create initially is what's called a mosaic pattern, so you don't get every cell affected, but this is just an eye example to make it visible. We injected something that would affect the eye, and it affects the pigment of the retinal pigment. And so a wild type, a normal fish would have an all black eye, and this injected one is speckled. So some of the cells have the normal pigment, and some of them have lost their pigment. So imagine if we're doing that in a gene affecting the brain, some of the cells have lost their function, some of them have not lost the function. And if you do this and breed these animals out, you can actually have a complete loss of function or a partial loss of function, which is quite relevant in our models. So we've got these tools. Which genes are we going to model? And as you know, there are lots of different genes relevant to epilepsy. We have at least 20,000 or so genes encoding for various proteins in our body, and many of them are relevant to the nervous system. If you do a simple literature search looking at the published research literature and the clinical literature, there are lots and lots of genes associated with epilepsy. So this is our epilepsy list. So out of our 20,000 or so genes, if you just do a search, so let the computer do the search for you, over 2,000 of them are somehow associated with epilepsy. This doesn't mean that these genes uniquely cause epilepsy and nothing else, but there's some association. So every one of the genes that Sarah mentioned and that we'll talk about have an association with epilepsy. There are other conditions where there are many, many other things going on as well, and epilepsy can be part of those conditions, but this is the gene list that you get. 22,000 genes is a lot of people time in terms of if you really wanted to systematically go through them. And we thought, well, let's figure out which of these are really going to be the most important. And there's a vast amount of genetic material available now. The reason we have these gene discoveries is in part we've had this Human Genome Project. There's been a huge revolution, not just in neuroscience and in epilepsy, but in medicine in general. And so we've got lots of tools in bioinformatics and lots of genetic information available. And so it turns out that about 120,000 people's sequence data are available. And so we can look at some of these genes and say, okay, this gene is this big, I'd expect it to have 10 changes across a room this size. And we'd look at all of our sequence and say, gee, nobody in this room has a change in that gene. That gene doesn't seem to like change. And so you can do that systematically, and you can put statistics to it, and you can do that to all of the genes now with all of these data. So we were able to go through this list of over 2,000 genes associated with epilepsy and say, well, about 300 of them actually don't like to be perturbed. So if you look at a gene like SCN1A or KCNQ2, you put any of our epilepsy-related genes that we know are strongly associated with epilepsy, they're out of the ballpark in terms of being able to not tolerate changes. And so that's a very powerful tool. We're looking now to say, let's look at a large group of genes and not just take the ones we know, but other ones in addition to sort of expand our knowledge base. We're down to about 300. That's still a lot. Now we know our model system is going to be the zebrafish. Let's see which ones are enough like humans that it's going to be worth the effort to model them. And there we get down to a little over 200. So that's a large list, but a tractable list for sort of a 5-year project with 3 or 4 people involved, sort of all hands on deck doing a project like this. We then said, let's focus first on the ones where there isn't other work going on, because if we're talking about the field of SYNGAP1, for example, Gavin and colleagues have sort of got that covered after years and years of research. There's really little point in our reinventing the wheel, if not just to have maybe a positive control. But there's some genes where there's not a lot of work being done, or where there's early work being done, where we really would like to try to advance the field in those areas. So we got down to a list of 77, which is sort of now closer to a list of 100. But a tractable list of genes we thought we could model. So I'm not going to show you the data from these yet. What I will say is that we were a little bit surprised, because it's still a relatively new field, how many of them would actually show us seizure-like behavior. And we were hoping for maybe 15 to 20%, because that would give us 15 or 20 models. It turns out about a third of them are actually showing us behavioral seizures. And so we're now going through the rather painstaking task of the EEG part, which is very low throughput, to figure out which ones we can validate. And there's all sorts of imaging ways to do that too. And then we're going to hone in on those models. So that's still a work in progress. And maybe if I'm back in 10 years, I'll have data to fill that in. But I wanted to really now transition to say, well, we have all of these exciting things going on in the zebrafish field, lots of exciting work you'll hear about in cell culture work and in mouse models that you're going to hear about. But what can we do today? So imagine me in my clinic on Tuesdays, and I've got these great, exciting experiments going on across the street and in the basement, and that really sort of drives the whole enterprise of a translational epilepsy genetics program. I still have a patient in front of me, and I would still like to do something useful. And I'd like to do something that's not just useful in a general sense, but really takes into account their genetic epilepsy and their specific diagnosis. So what can we do? It turns out we can do a lot. It turns out we can do quite a lot by incorporating the research into our practice, and this is actually an area where you all come in. And I'm just putting genes here, not to be excluding other genes, but genes where we've had some active interest and some involvement. So certainly Dravet syndrome has been one of the first out and one of the first communities to organize, and a lot of the different groups have gotten together, and there have been various registries and so on, but we see this often enough, and there are clinical trials now for Dravet syndrome, that we have a reasonably well-organized group. We started an American registry for patients with variants in PCDH19 and have been partnering with others in the community who've done the same. We're very, very pleased that Scotty's group, KCNQ2 Alliance, has been partnering with us to start a pilot natural history study, which we hope will grow to a broader natural history study. And we're quite excited that there's an Australian partnership here with Ingrid's group and colleagues. And then speaking of Ingrid's group and colleagues, the SCN2A community is quite organized as well, and there's hope to be able to bring that together, and we're hoping that we can contribute to that as well. So a lot of activity here, and again, this is where we are going to go from one patient at a time to groups of patients and really bringing clinical science to this. Gene discovery is still happening, and that could be another whole day's symposium as well, but we're still learning about new genes, and as we learn about new genes that we think are brand new and we didn't think had anything to do with brain development or epilepsy, it turns out that everything's sort of coming together, and we're sort of honing in on core processes that are important. So we're still learning a lot, and actually that I think is going to drive not only our ability to diagnose patients, but our ability to sort of prioritize which genes and pathways are important to study in the basic lab. We're studying PCDH19 among other genes within our many gene fish in the zebrafish. I'm also partnering with one of my lab neighbors on a DEPDC5 mouse, and I won't talk about this gene anymore, but I'll talk about the pathway. Our group and others are studying many, many genes in the iPSC, the induced pluripotent stem cell models. We're looking at KCNQ2 specifically and are waiting on a funding decision to be able to do something with many samples that we were able to obtain from a recent KCNQ2 conference. So we're crossing our fingers for that. And then we've got a small group of patients families whose children have SCN8A mutations, where we're trying to study those in the dish to see what might be useful for treatment, much of which has been done by other groups. So that's our little microcosm in terms of what my group is doing. You'll hear more from what other groups are doing. So again, that's the research side, but what about the treatment? We still want to do something for these kids. And how can we do something today, but also look towards the future of trying to do something better systematically? And so what are we looking for? Well, we want them to work. And what do we want them to work for? And these may seem like obvious questions, but when we really are thinking about, well, what do we want to look at in these natural history studies, we have an eye towards this. We want to look at seizures, of course. We'd like to look at EEG activity if there's evidence that it correlates with seizures or developmental and cognitive outcomes. Cognitive and behavioral symptoms, in some cases movement disorder symptoms. There's a whole host of things that are important, and we have to sort of decide as groups and as mini groups which ones do we really want to focus on, and what are our main goals for treatment and what might be our secondary goals for treatment. Then when we get down to my Tuesday afternoon, what can children tolerate? So we can try to target specific dysfunction, but we really want to avoid unneeded side effects, and you all know this from your lives with your children. We can make many, many seizures go away with powerful medications that can make any of us sleep all day, and that is really not the purpose, right? We really want to have our patients, our children, alive and vibrant and doing the best that they can, and so the treatments we would hope wouldn't make them worse. We'd really want them to make them better. So really what this comes down to is a benefit-risk balance in terms of not just tolerability, and then of course we have medical safety. So there's a lot of things to juggle. And I just want to say one word in favor of clinical trials, because I'm going to tell you about some N-of-1 experiences. But really, when are clinical trials appropriate is really as often as we can get them. And the power of organizing as a group is that we can actually generate enough group power to have clinical trials together. And that, I think, will get us to a really more scientific approach to treatment than what we have now. But we have to do something now. And so it turns out, and this is something that we have to bring up in the US a lot in terms of justification for why do we want to do genetic testing. You may all think, why would anybody even ask, why did you do genetic testing? If you have a developmental epileptic encephalopathy, isn't it obvious that you do genetic testing? And it seems obvious to us in the room, and it seems obvious to the scientists of us in the room, but it's not necessarily obvious to our healthcare system, and it's something that has to be really sort of moved forward. Some of you all are involved in the moving forward. There's a new effort by the SCN2A and KCNQ2 communities to do earlier genetic testing for very, very young children, because those tend to be genes that are seen very often, and that's a really important piece that we're pushing. My neonatology colleagues and my genetic colleagues are doing this in our NICU. They're trying to enroll patients to show our healthcare system that this is important. But so why is it important? I'm not going to go through all of these genes. I think other than CDKL5, which has its own whole set of treatment finesse, most of the genes in the room are represented, except for SYNGAP1, which I'll talk about. Some of these actually have some observations. Generally speaking, we try not to use dilantin lamotrigine for Dravet syndrome for SCN1A-related epilepsy, although it's a generality. There are trials for some of these that I wanted to mention. And some of these are metabolic syndromes where you actually have deficiencies that you might be able to replete with oral treatments, and so that's important as well. So we have potentially treatable causes where today we are changing our management based on a genetic diagnosis. I'm going to give you 3 quick vignettes on some of these. One will be on a gene called CAD, quick vignette on SYNGAP1, although with very generic approaches, and then a little quick story on focal cortical dysplasia. So I'm going to start with a little vignette on SYNGAP1. So this really reflects an experience I'm having now with one of the patients in my clinic who was transferred from a colleague's clinic from down in Philadelphia. And this is really a way, this is not precision medicine. This is sort of trying to target treatment when we don't yet have precision medicine and hope that precision medicine will come. Now this was a child who I saw at a little over 2 years of age. He had had developmental delay, and that's what actually prompted his initial genetic evaluation. He then started having seizures that consisted of eye fluttering and head drops, and had a generalized spike in wave abnormality on his EEG. He had neuroimaging that was normal, and he was then found to have a truncating variant, so a change in the DNA that stops the protein short when it's being made. And he'd had inadequate responses to a couple of different anti-epileptic medications. He was on topiramate when I first saw him. And the question was raised, well, if he's got a premature stop codon, why not address this rather than just continuing down the list of generally used anti-epileptic medications that, the most finesse we would have would be, well, it's a generalized epilepsy. Let's look at least at medications that work on generalized epilepsy. And at the end, we may just go back to that. We may have to say it's a generalized epilepsy. Let's just use those types of medications. But this is where we were. And so it turns out that there are some medications that have been used to try to modify this. And so there are actually medications that are now used in trials in Dravet syndrome and have been used for years in cystic fibrosis and Duchenne's muscular dystrophy and other conditions where if you've got a premature stop codon, you're trying to make a protein and all of a sudden instead of ending here, you've got a stop sign here and you cut your journey short, that you try to be able to read through it. And there is actually a nicely worked out set of potential molecular mechanisms. It may not be, in the textbook it looks pretty clean. It looks like it ought to work. It may not necessarily work as nicely in our bodies and in our cells as it's supposed to. So I'll leave that as a disclaimer, but it's a rational approach. And so this little boy, whom I'm now following, when he's seen by my colleague, had whole exome sequencing results that gave him this results back in March of 2017. His father gave me this timeline, and the family with the doctor reached out to the company that makes this drug, Adalurin, which can result in reading through of these early stops. And it really wasn't an option at the time to get the drug from the company unless you had Dravet syndrome and could enroll in the Dravet trial at that time. For a while, they tried azithromycin. Azithromycin, which is an antibiotic, and gentamicin, an antibiotic, had been tried in other conditions to do this read-through. They thought it would be worth a try. It didn't really do much of anything, so they stopped it on the recommendation of their physician. And then they transitioned up to Boston, and we said, let's just see. We'd been hearing a little bit more preliminary data from the Adalurin trials from Dravet. Maybe the company will be interested in supplying it now. So we reappealed to the FDA and the company, and they were. And they said, the company said, if the FDA is okay, we're okay. And the FDA said, well, give us some information from the company. And my wonderful team was able to sort of negotiate all of these different exchanges of lots of huge documents. And finally, we actually had FDA approval for investigational new drug use for Adalurin in this condition with a truncating variant in SYNGAP1. And then our own local regulatory group, our IRB, approved it for just specifically for the use in this child. So we've started it. So this is not a clinical trial. It is not blinded. It's an N of 1 experience. And I've thrown that as a word of caution because anytime we start something new, whether it's for epilepsy or for development or I have a headache and I'm going to take my Motrin, those first few minutes when I've just ingested it and it's still here, the headache started to relieve itself. And we know that there's a power in belief. And we know when we're starting something new, we'd like it to work. And so we do need to have some objective things to look at. And so what we're going to look at now, on the child's 3rd birthday, he happened to be in clinic, because that's when we could see him. They signed consent for that. How are we going to look for progress? So we'll look at seizures, which we do with seizure diaries. And you all can tell us, as well as anybody can tell you, that there's some objectivity there, but it depends on who the observer is and whether they know there's a change in treatment or not. We're going to look at EEG, and we're going to do formal developmental assessment. So this is very much a work in progress, and we have some marginal hope that maybe there'll be some improvement, and if there isn't, at least we've tried it. I think the challenge for me, not as the scientist, but as the doctor in this situation is, when should we move on down the list of other good old generalized epilepsy drugs that might be applicable here? And that's sort of what we're weighing in terms of the pros and cons of doing this. The downside of being on the drug, not so much. The side effects are really minimal, if any. But the downside of doing that to the exclusion of other standard treatments might be that maybe we're not letting him have the relief he might otherwise have. So it's not an easy situation. And again, it's an argument for, doing safe things is fine, but doing them in a trial would be better. A quick example of another situation like this is a case of 2 siblings who came up from Florida. They were known to a nurse who is a friend of a friend, and she said, they've got these variants in GRIN2A, can you see them? And so I said, sure, you can see them. And by the time they had booked their flights and we had the visit set up, we got the testing back, and it was actually a maternally inherited variant. And we said, yeah, maybe not so much. And the variant itself didn't look so compelling, but wait, there's an exome. And the exome report was handed to me while the child and the mom went to the restroom, and I had about 30 seconds to look at it. And we said, oh, this is interesting. I've never heard of this gene. It's a gene called CAD. It encodes a protein that turns out to be quite critical. And so our DNA and our RNA are made of these purines and pyrimidine, these basic building blocks. And as you might imagine, if you don't have those basic building blocks in your cells, it's very, very, very detrimental. And the condition that arises from not being able to produce these pyrimidines is quite devastating. The older sibling in the family had early recognition of delays, early onset of seizures, status epilepticus, and had severe consequences, was in pharmacological coma for months. And it's not doing well at all. And then the second child who was affected in the family had some mild delays noted early on, seizures that are about the same age, got worked up right away and was found to have similar patterns, including this little area of the brain here, the cerebellum, which was starting to shrink on the MRI. And along with that, her cerebellar function was suffering, her walking became more wobbly, and then as she started having more and more seizures, dozens a day, her minimal speech actually went to no speech, and she was really very minimally interactive. And so things were going downhill very quickly at the time that we met them. I'm not going to go into the details. We absolutely can look at this in the zebrafish. We absolutely can have the fish seize, and we absolutely see that there's a survival deficit. So there's a lot we can do in the lab that we're working on in the lab. But around the same time that we were seeing this family, this paper actually hit PubMed, the literature server, and it turns out that a group in Austria had actually looked at these children, and when you're not making enough of these pyrimidines, it turns out that some of them are available as health food supplements. So good old health food stores have uridine. So we said, this is not very scientific at all, and how are we going to get this? And we found a pharmacy down in Florida that would compound it, but how much do we use, and what dosing range do we use, and how do we assay this and see if we're doing anything good, and how do we make sure we're not doing any harm? So it turns out there's a lot of people who are on uridine for all sorts of reasons on the health food websites. And the rationale for actually doing something now, rather than waiting for the 3 years it's going to take for the experiments to happen in the lab, is the following. We had diagnostic certainty. We had 2 siblings. They each had a variant from each of the parents. We had actually good bioinformatic data to let us predict these were actually damaging the protein. The MRIs were progressing, not only in the boy who was already severely affected, but in the sister who was just beginning to show symptoms. Her MRI was already on its way to not looking good. We also had a really nice non-nervous system marker, which is this long word, anisopoikilocytosis, which basically means that the red cells are all funny looking in different sizes. And the red cells, as you can tell, are pretty easy to assay, so we don't have to necessarily go looking at details in the brain. We've got a nice assay in our arm, wherever we want to take blood from. So we had diagnostic certainty. And the risk of not pursuing treatment was that her seizures were going on relentlessly. We were really worried she'd go into status epilepticus and have the same consequences as her brother, and her development was going downhill as well. So we had sort of clinical urgency. And it turns out that there's a theoretical risk of anybody having an allergy to medicine, and anything new you have to take with a grain of salt and also with a bit of caution. There were no known downsides of being on uridine. So together with her local neurologist and their geneticist and a whole team of us together, we said, we're going to try this. We're going to use the dosing that the Austrians use, and we're just going to give it a go. I cannot tell you it was just because of the uridine, but I promised the mom I would show you all this. I just saw them a couple weeks ago in clinic. And this is not sort of a staged before and after where it was a bad day and a good day. And I've actually seen the child in both. This child was like, I couldn't, I had to wash my hands about 10 times in the room because she's so persuasive with her commands, verbal and nonverbal, that she's just so engaged. Completely different child than whom we saw running, walking, getting some words back now. And so I can't stand here and tell you that this was 100% this uridine because lots of other things happen. Real life and real medicine is that you change a lot of things. And so other meds had been started over the summer. Meds that might have been causing toxicity were removed. And so this is a very, very messy N-of-1 experience where, at least for now, knock on wood, the end result is that we're making some progress in the right direction. So what can we do objectively? Well, we can at least look at some big things that we were hoping would go away that had been persistent. So she'd been having a lot of tonic-clonic convulsive seizures that have not been present for the last 10 months since starting the medication. Her drop seizures actually didn't go away right away, but with increasing the dose of this and other meds, they're better. So I'm not sure if we can claim credit for that. She still has some brief absences that I saw in the office. They're not gone yet, so we've got to track those. Her language is improving. My wonderful Harvard Boston Children's Hospital has lost the blood smear, so my hematology colleague, as of this morning, still couldn't find it. But we are tracking the blood, how the red blood cells look, because you'd like to. Our red blood cells actually turn over every 4 months or so, and so if we're making a difference with treating the biological abnormality, the red cells should look better. So from last summer to this fall, they looked a little bit better, but there were still some funny-looking cells. I wasn't satisfied we were giving the maximum dose we could. We went up on the dose. We've checked the blood smear again. They're looking in the cabinets. So hopefully we'll get that. And the other thing is we can look objectively at the MRI. We know the progression of this disorder as the cerebellum gets smaller and smaller and smaller. We can see if we've stopped that as well. So objective things to look at. The last example I'm going to give you really, really quickly is, this is the example that took me from being a full-time doctor to being a mostly researcher and sometimes doctor. And this was a condition that really takes the rare to the common. We looked at large brain malformations. We looked at them in the microscope. We thought that maybe they were due to mutations that happened early in development that affected just a small number of cells, but all of those cells' offspring would go through the brain and create these little patches of focal epilepsy. So this, it turns out, is really common. We see this every week in our epilepsy practice. And we didn't know anything about the genetics. And when I was in my epilepsy fellowship, seeing this in the operating room and seeing it in the microscope, I thought, well, gee, I've been, in the trench doing my residency. Maybe I missed the genetics of this. Clearly, we must know by now what's causing it. And we didn't. And Fast forward now 15 years, it turns out we actually understand a whole lot of it, and this is work done by a wonderful graduate student, Alyssa DeGama, in Chris Walsh's lab upstairs from me, where I've been working in partnership for many years, and it turns out we can now figure out about 40% of them. And so we went from absolutely nothing until 2012 to about 40% of these cases solved. And not only are they cases solved, but they're cases solved in the very same pathway as tuberous sclerosis complex, very same pathway that I mentioned earlier, this DEPDC5, which Ingrid and others here have really put on the map, not for this particular type of epilepsy, but for a much more common epilepsy. And now when we talk about collaboration across the globe, and we've looked at 4,000 people with epilepsy, it turns out that this is one of the hot players on the gene list of genes involved in epilepsy, even for people who don't have a family history. And so all of our work on the rare is converging, and it's really informing the common. And so I think when we learn how to treat this, it may actually not just help our once-a-week patients with these disorders, but actually a much more vast group of patients. And so this really does provide a translational medicine opportunity. This is Dante, my first patient who had a variant of this pathway that I found by grinding up his brain tissue, bringing in getting in a blood sample. And when I told his mother, who's a science teacher and now a principal, that we'd figured it out and it was a somatic variant, she said, "You told me that years ago." I said, "Well, I knew that and I believed that, but I've shown it and it's published now." She said, "That's nice, but I believed you years ago." So she said, "What are we going to do about it now? We've already done a hemispherectomy," which is true, but we hope we can actually, with the next generation of kids, do something better. I don't want to go over. We have a lot of challenges. This pathway is very, very complicated, as you might imagine. I've sort of kept things at a high level to cover more breadth, but there's a whole depth here, and this is decades and decades of work that needs to be worked out in terms of how to do this in a refined fashion. But there is hope, and we've got real diagnoses now that we can work on. So I just want to close by saying that there's tremendous power in partnering. When I was in school and in residency, we didn't have conferences like this where doctors and scientists got together with families, and I think it's a very powerful thing that you're doing. It lets us, not only see our patients in our clinic, which is, as you know, sort of, fixed and rushed and sterile and so on, but really get to sort of see you out in your element, and you get to see us out in our element. I think that's very valuable to be able to connect as people. So thank you, and thank you, Geta, and I just want to thank my team. We had a one-time visit from guest Professor Sheffer. My team was completely agog because Ingrid came over and she visited the lab and saw the fish, and so that was quite exciting. None of this happens without mentorship, none of this happens without international collaboration, and none of this happens without community. And so I'll just tell a quick story. The first exome I was able to do in my research lab was through a $1,000 donation, which was the entire fund of the Aaron Zodahara group. This was a group of a few families who had a basketball tournament to raise money for us to do exome sequencing. DSF picked up on that and said, "Why don't you apply for one of our grants? Maybe we can do a few more." I said, "But this is Odahara syndrome." They said, "Yeah, we were that syndrome without a gene until someone found it. And so why don't you find the Odahara genes now?" And so that has just led to a wonderful ability for us as physician scientists to engage with the community, and that really keeps the research alive, not just for this group here, but for now for our group that has just grown to be the size of an ice cream truck, and has really brought a lot, I think, to our own ability to not just see one patient at a time, but groups of patients and scientific questions, not as separate things, but as one thing that we really have to put together. So thanks very much for your attention.
Stay. Oh, great. That was fantastic. We want to allow time for questions. So Chris is going to go around with the microphone, as is Danny. Does anybody have any questions you would like to ask Dr. Poduri? All right, we have a couple. Oh, that's for others. Okay. Does anybody have any questions? Yes.
Sorry, yep, this is working. I just wondered whether you'd used anything like the CRISPR technology, and I don't know, obviously I come from a bit of a science background, but not anywhere near your level, so I just wondered whether that, I had read that UCLA Brain Research Institute in America is doing a lot with the CRISPR, and they claim sort of to be able to potentially, well, they're doing studies in mice at the moment, and they're able to reverse brain disorders such as Rett syndrome and X-linked other pathologies like that. And I guess that's directly comparable, a little bit more comparable to our situation, which we have a little bit more of a rarer called IQ-SEC2. And so it's an X-linked dominant de novo condition. And yeah, I just wondered whether you could comment a little bit about the CRISPR.
Sure. Yeah, so that technology, it's really sort of in some ways, sort of a beautiful, simple technology that was really discovered to be present in species that are not human species, but we were able to sort of take that ability for enzymes to go in and edit the DNA. So you can sort of imagine that CRISPR technology as a DNA editing strategy. So what's been possible, and this can be done in cells, this can be done in organisms, this can be done in patient-derived cells, and then you can actually go into the cells in addition and try to fix the variant that's actually there. And basically, you can think of it as if you've got your DNA written out, as a little sort of pair of scissors. And so if you just threw a bunch of scissors at a bunch of strings, you'd get a bunch of cuts wherever. The CRISPR actually is a way of doing it in a targeted fashion. So you've got your DNA, imagine all these strings, and you're sending the scissors to a certain spot. And you're saying, cut that spot. So if I want to make a model, let's say I wanted to make an IQ-SEC2 model, I would say, well, I want to cut that spot. I want to remove that gene, and let's make a model where there's a loss of IQ-SEC2. So that's quite doable, again, in any number of different systems. So we can do that in a fish. It takes longer. We can do it in a mouse. You can also make that in a dish of cells. You can also then take, and this requires a little bit more refinement, because if I just want to make a cut to cause dysfunction of the gene, that's kind of step one. If I wanted to make a specific cut, let's say I wanted to make a specific cut that would approximate your child's variant, that requires a little bit more refinement, but that now is also within reach. I mean, these are technological advances that were sort of science fiction 10 years ago, but have really come a long way. And so all of our big institutions where we do these sort of works have genomic cores and people whose job it is to think about how to get this to be as precise as possible. So it's very much coming. I think the important question is sort of how do we go from there back to the patients. And so, you know, the best illustration that I've seen of this now is, and these are from not necessarily just epilepsy studies, but people studying ALS and other neurological disorders, is you take cells, you make them pluripotent, you make them into neurons, and you can study them. And we often were sort of comparing them to other cells of people who weren't affected with illnesses and so on. But it turns out that experimentally, if you want to say, well, I want everything to be the same other than just that one mutation, that one variant, well, what if you could just go in and send the scissors there and fix it? So people have been doing that to sort of create controls. And that led to this idea, well, if we can do that to create a better scientific experiment, well, what are we doing here? We're fixing the defect. Can we do that in a broader way and deliver back the normal cells? And so that's one line of logic that people are testing. And there are lots of other molecular strategies that people are testing. So I think it went from really something we'd never heard of several years ago to something we'd heard of, but we weren't sure how fast it would take off, to really a very, very powerful technique. You know, how soon it goes from these experimental lab paradigms to really getting to patients, I think that's what we have to keep asking ourselves and pushing ourselves to say, when are we ready? When is this not just potentially effective, but safe to take back to patients? And how will we do that? And how will we know that we're on the right track? What are we going to measure? And so those are all the next set of questions we need to ask.
Great question. Do we have any other questions? We'll probably have time for one more, and then we're going to have a Q&A with all our professors at the end of the day. So if you didn't get a chance now, there will be breaks, and we'll have that time at the end of the session today. Another question? We've got time for one more. All right. Well, may I just say one more thing before I let Anne go? I talked about how amazing Anne is, and you got an opportunity to see all that kind of scientific heft and the passion that drives it. May I also point out that when we had our KCNQ2 conference, KCNQ2 Cure conference in the United States last year, and we had it in Boston to be connected with Boston Children's, Anne not only was doing all the stem cell collection and all of that, she drove the car so that people could get from our conference to the lab so that they could have their blood taken, because it all fell apart, and she jumped into the breach and actually drove the car. So when I say Ann Poduri is a very special scientist and an incredible woman, please join me in giving her a great round of applause.